Let us talk about the primary structure of protein. Now, if I am to write the exam and they ask, explain the primary structure of proteins with relation to its biological functions. Now, what are you going to say for the primary structure? This is what I gave here. I said the primary structure of a protein is the sequence and number of amino acid. This is supposed to be amino acid in the polypeptide chain. Each protein has a unique sequence of amino acid which is determined by the gene contained in the DNA. The primary structure of a protein is largely responsible for its function. Primary structure is what is responsible for the functions of the protein. A vast majority of genetic diseases are due to the abnormality in amino acid sequence of protein. For instance, sickle cell anemia, which is due to the change of glutamic acid in the beta chain to what? To valine. So only one of the amino acid, glutamic acid, is changed to valine in sickle cell anemia. So instead of the normal HBO, they are now having what? HBS, which is the sickle cell anemia. Is that understood? So that is what the primary structure. It talks about the sequence and the number of amino acid in the polypeptide chain. Each protein has a unique sequence of amino acid which is determined by the genes contained in the DNA. The primary structure of a protein is largely responsible for its function. A vast majority of genetic diseases are due to abnormalities in the amino acid sequence of what? The primary structure. Now, another thing that you need to know is that the primary structure is the backbone for all other what? Structure. For instance, the secondary structure is formed as a result of the folding and the twisting of the primary structure. So if the question now comes that what is the secondary structure of protein with relation to its biological function, you can now say that the secondary function of or the secondary structure of protein talks about the folding and the twisting of the polypeptide chain. Are we getting it now? Resulting to either the alpha helix or the what? Beta plate. Beta pleated helix or beta plates, alpha helix or beta plates. Now, talking about the secondary structure now, if you want to write the biological function, or let's say its biological function, you relate it to primary structure. You now see that since the primary structure is the backbone of all, a change in the sequence of amino acid affects the what? The secondary structure of the protein. Then go to tertiary. Tertiary talks about the 3D arrangement of what? Of polypeptide chain, 3D arrangement. Why the quaternary talks about the relationship between two polypeptide chain. But in most cases, they usually ask about the primary structure. If they say tertiary, it talks about the 3D arrangement or the spatial arrangement, 3D arrangement of proteins. So that is that for the primary structure. Now, name and draw example of each of the following amino acid. One example, aromatic. Example of aromatic amino acid is what? Is tyrosine. Let me draw the structure of tyrosine. Tyrosine is something like this. All those that have the benzene ring are all aromatic. An example is tyrosine. Now, this is the structure of tyrosine. You have C, H, and then you have this group here is, let's say, C, this CH2, then this group continues to form C, and here is OOH, here is NH2, and here is H. This is tyrosine. Now, another one is phenylalanine. That means phenyl group with what? Alanine. That is structure of phenylalanine. Now, it depends on how you've learned it before. If you've learned your own structure, you may not need to cram my own because there are so many ways that these structures can be drawn. You have H2, you have C, you have NH2, and then you have hydrogen and COOH. This is phenylalanine. Phenylalanine. And then this is tyrosine. These two are aromatic amino acid. Let's look at the next one. The next one is amino acid. Example of amino acid. The only example of amino acid is what? Proline. So you should draw a name. Tyrosine, phenylalanine. Now, the only example of amino acid is what? Proline. And the structure of proline is this. You have something like this. There you have this. You have this. You have hydrogen. And there you have COOH. This is structure of proline. Whichever way you want to draw it. Now you can decide to draw it like this. So long you know that there is H here. And then you have COOH. This is proline. Are you getting it? Anyhow you want to draw it. So long you show 1, 2, 3, 4. Then COOH. And this one making it 5 is proline. So whichever way you want to draw it is also the structure of what? Proline. I prefer it this way. I think I prefer it this way. Or you may want to bring the COOH group out to now give something like this. Now, if this is the ionized form, what will it now be? It will now be H2 here. There here will be what? Positive. And then you now have COO minus. If you want to put it in the ionized form, because if this one loses a proton, it will add here. And this one now will now be positively charged. So you have NH2 plus and then COO minus. So that is the amino acid we have. Now let's look at the next. Sulfur containing. Example of sulfur containing amino acid is example of sulfur containing. Why am I forgetting? Cysteine. So we have the structure as this. Cysteine is COOH. There you have NH2. And there you have it like this. And then you have CH2 and then SH. This is structure of cysteine. Then we also have methionine. You can say L-cysteine. This is the L form. You saw how I drew the configuration. This is the L form. Then you can have methionine. And then this is CH2, CH2, S, CH3. This L-methionine. All these are sulfur containing amino acids. Now let's look at the next. If the extinction coefficient and path length of BSA, now the extinction coefficient is E, 43,000, depend on what is given to you here. 43,834 mole per centimeter, and then the path length L is 1 centimeter respectively. Calculate the concentration C if the absorbance A is what? 0.550. Now what is the formula? Using Beer-Lambert's law, is A is equals to what? ECL. Where E is the extinction coefficient, C is the concentration, and L is the path length. So making concentration subject to the formula, what are we going to have? To make concentration subject to the formula, we have A is what? ECL. To make C subject to the formula, divide both sides by what? EL. EL cancels EL, and C will be what? A all over EL. Where's the absorbance? 0.550. What is E? Is the extinction coefficient. The path length is what? 1 centimeter. Now, recall that this per mole per centimeter. Are we getting it? Then we have EL is per centimeter. These two, we cancel this, what you have here. So you have mole. Are we getting it? At the end of the day. So our concentration now will be equals to divide this with this. And then what you have is your answer. When you divide that, it's going to give us, time has gone far, 0.550 divided by 43,834. And this will give us 0.00004 125 molar, and that is the solution to this question. Okay, now you don't need to use this value. It just telling you the wavelength at which it absorbs, 0.55. 0.55 divided by anything you have here. Now you don't need to convert this to meter because centimeter and this. So if the unit here are the same, you don't need to convert. And this is your answer for concentration. Now let's look at the next. Write a short note on DNA, deoxyribonucleic acid. Deoxyribonucleic acid is a polymer of nucleotide containing a deoxy sugar containing a 2-deoxyribose sugar containing a 2-deoxyribose sugar. It also has nitrogenous bases such as adenine, guanine, cytosine, and thymine. The nucleotides in DNA, the nucleotides are joined together. They are joined together by a phosphodiester bond. Phosphodiester bond. Okay? It usually has two chains that runs antiparallel to each other. And are joined together by hydrogen bond between adenine and thymine, and then cytosine and what? Guanine, or guanine and cytosine. Now you saw what I did here: 2 hydrogen bond between adenine and thymine, and 3 hydrogen bond between guanine and what? Cytosine. It is responsible, or most genetic materials are stored in the DNA. Most genetic materials are stored in the DNA. This is not something that is new to us. Most genetic materials are stored in the DNA. Then at least if you do this, you've written a good note on DNA. Is that not so? Deoxyribonucleic acid. Most genetic materials are stored in DNA. It usually has chains that runs antiparallel to each other and are joined together by hydrogen bond between adenine and thymine, and cytosine and what? Guanosine. 3 hydrogen bonds like this. Is that okay? So you may have given the Watson-Crick model of what? The DNA. Watson-Crick model. I said that it has two. Where is it? So that the nucleotide are joined together by phosphodiester bond. It has two chains that runs antiparallel to each other, and the two chains are right-handed helix. Okay, so that is that. Now, draw the structure of a named purine nucleotide found in RNA. Draw the structure of a named purine nucleotide found in RNA. Now the purine, let me start by saying nucleotides are of two types. Am I saying nucleotides? the base, nitrogenous base. Let me start by saying nucleotides comprises of this. A nucleotide comprises of nitrogenous base, phosphate group, you have the ribose sugar, Now, the nitrogenous bases are of two types. You have the purine base and then the pyrimidine base. Now, under the purine base, you have adenine, and guanine. Now, adenine is called cis-amino purine. So, if you want to draw a named purine found in only RNA, let's use adenine. Let's say adenosine tri or diphosphate. Adenosine diphosphate, which is ATP. Let's not use triphosphate, let's use monophosphate. Adenosine monophosphate. So we come here to draw the structure of adenosine monophosphate. All you need to do is adenosine monophosphate. Now, since they said is in RNA, this is what you are going to do. First of all, let's draw the ribose sugar. The ribose sugar is something like this. OH, H, OH, H, now, like I said, you first of all draw the ribose sugar, which is something like this. Get out. With something like this. These are the ribose sugar. Now, note that the ribose sugar is for RNA. So it means that carbon number 2 will also have its OH. Are we getting it? Now, if it is for DNA, there will be nothing there. Now you have OH here and H. Now, this group here will contain the phosphate, which is monophosphate. You have hydrogen here. Now, this group here is supposed to be CH2OH. But because of the phosphate group, let's write it in such a way that it will face this other side. You write it as CH2, then O, then you are now going to put P, double bond O, O minus, and what? O minus. This is how the phosphate group attaches to the ribose sugar. Now, adenosine means you draw the structure of what? Adenine, and then add it to it. Now, the structure of adenine, I said adenine is cis-amino purine. This is the structure of adenine. You have something like this. This is N, double bond, something like this. Then you have this group here. This is nitrogen. Then this group here. This is also another nitrogen. Then the double bond will be formed here. Okay. Now, the structure of adenosine monophosphate. Now, adenine is cis-amino purine. It means that there is amino group here. I told you that the numbering for this one, start 1, then go round, and then come back to this place. That's how to number the ring. Okay, the ring numbering start from here. 1, 2, here is 3, here is 4. Think I'm making mistake. This is 1, here is 2, here is 3, here is 4, here is 5, and here is what? 6, 7, 8, 9. So you call it adenosine, which is 6-amino purine. Okay, 6-amino purine. So you come here, this one, it usually draw the structure of a named purine nucleotide found in RNA. Let's say this place is pyrimidine, not purine, found in RNA. It means you have to draw the structure of either uracil or what? Cytosine, and attach it here, because thymine is only found in DNA. So what is the structure of uracil? Now, uracil is a pyrimidine base. So you can add uracil here. Now the structure of uracil is this. You have your nitrogen. You have your nitrogen here. And then here you have double bond O. Here this group adds here. Then you have this group here. And then double bond here. Of course, you add one hydrogen here. So, for the pyrimidine base exclusively found in RNA, this one now, you now say the name is, the structure we draw is for uracil. So you say this is uradine 5 monophosphate. Now, if it is the structure of cytosine that you put here, let's assume it's cytosine. This is the structure of cytosine. You have N. Let's say it attaches here. You have this. Now, there you put this NH2. You also have your double bond here. Then you have double bond O here. And then you have this. The name now will now be what? Cytidine 5 monophosphate. So the most important thing you must take note of is, if it is DNA, what do? We remove this OH from here. Is that okay? Now, if you add another phosphate group, you can decide to add another phosphate group until it continues like that. What are you now going to call this? Diphosphate. Now, add another phosphate group minus. What do you call it? Triphosphate. And it continues like that. So that is that for DNA and RNA. Now, draw the structure of hyaluronic acid. Hyaluronic acid consists of glucuronic acid and N-acetylglucosamine. This is the structure. Please know it because it's going to come out. You can pause it and then review it together. Then chondroitin 4 sulfate consists of glucuronic acid and N-acetylgalactosamine 4 sulfate. This is the structure. Now, for heparin, consists of D-glucuronate 2 sulfate and N-sulfoglucosamine 6 sulfate. You can decide to pause it and then memorize the structure. And then, of course, we have dermatan sulfate. And then finally, keratin sulfate. So let's take note of all these dermatan sulfate, keratin sulfate. I believe that is all. So please, you can pause this particular part and then review them together, okay? Try and memorize the structure. It's very important you know them. So that is all for those past questions, apart from those ones that have to do with kinetics. I don't believe that questions on kinetics will come, but I don't know. You can review it in your spare time. But I would advise that you use that time and focus on the things that we actually taught on enzymes, like the classes of enzymes, the features of enzymes, the factors that affect rates, and those liver enzymes and heart enzymes. If you focus on those ones and not the kinetics and the rest. So thank you for your time. It's really late in the night, into the night already. I wish you success in all the exams.